this week started. This week we're joined by Dr. Kelsey Lucas, a collaborator of hers, and some of her graduate students. And just to start with a little bit about Dr. Lucas, she did her PhD in organismic and evolutionary biology at Harvard University, and there she studied the flexibility and swimming performance and fish swimming force production. She also did a postdoctoral fellowship at the University of Michigan, where she studied or learned about understanding the impacts of climate warming on fish physiology and understanding the links between fish morphology, some environmental characteristics, and habitat occupancy in Michigan inland lakes. And she's currently an assistant professor here in the biological sciences department. Some of the things that are in her future plans are she's, I think, currently renovating her lab, which is close to being done, and looking forward to getting some new equipment up and running. And some of her hobbies include long distance running, gaming, and baking. So thanks for joining us, all of you, and we're looking forward to your presentations.
All right, thank you very much for the invitation back to HPL. It's been a couple of years since I've done a seminar here, and the last time it was on Zoom. So it's so much more exciting to be doing this in person. I'm Kelsey Lucas. I'm a faculty member in biological sciences, and presenting with me today will be my close collaborator, Heather Jimninsky, as well as two of our students, Sarah Perry and Duncan Kennedy. Over the last few years, Heather and I have found that we have a lot of overlapping research interests and a lot of very similar questions in our approaches. And so while I've been waiting for my lab renovations, we've developed a lab scenario where we more or less are co-running a facility at this point. And so today we'll be telling you about some biomechanics and ecology things to understand fish distributions and evolution. And I want to start with a big thank you to many of the folks who have helped with all of this. As we are multiple lab groups, there are quite a long list of colleagues and collaborators and former students. I especially want to shout out Aspen Kozak, whose master's work will feature heavily in Heather's portion of the talk, as well as Heaven Duclos, who the former student of Heather's, who has helped with pretty much everybody's morphology work that you're going to see today. And so just to kind of set some context, this is the Human Performance Lab. So I'm here to talk to you about fish. So let's just set the stage. To understand life on Earth, we do need to understand aquatic systems. Now, I like this picture of the Earth because it highlights just how much of our planet is aquatic habitat. You can see the Pacific Ocean there and only a teeny bit of land. If you were actually to calculate out the volume of habitable space on our planet, about 90% of the world's habitable volume is in aquatic systems. That contains a huge diversity of life, including half of invertebrate phyla, which are exclusively marine. There are about 35,000 species of fishes. That's more than every other vertebrate combined. And about half of those fishes, I also just want to add, about half of those fishes are found in freshwater systems, which is a disproportionate amount when you really think about how much of the world is ocean versus freshwater. Now, among our lab groups, we tend to think about things in this ecomechanical framework. And while many of us here would consider ourselves biomechanists, or at least biomechanics adjacent, we can all appreciate, and we all can appreciate, that there's been a lot of work trying to understand the physical mechanics of fish behavior and swimming, including some of my own work that I presented at HPL a couple of years ago. Increasingly, we are trying to think about biomechanics' role within an environmental context and its place in ecology and evolution. And so that led to this ecomechanical framework, where we're thinking both about functional traits, or those biomechanical traits, morphological traits, that are technically our bread and butter in the biomechanical world, but also simultaneously thinking about how these traits interact with each other, as well as with environmental variables. And so that's just what all of this FT and EV is. It's just all of these functional traits and environmental variables interacting with each other. Then we also are curious about how all these functional traits and these biotic and abiotic variables drive performance and hence fitness in a habitat and in our natural settings. And so we're really excited today to tell you about some of our ongoing work within this ecomechanical framework and what we're learning and where we hope to go from here. So first we'll have Sarah up to tell us a little bit about some of her functional morphology work. And then we'll have Duncan who will come up and talk about some of his morphology and swimming work. Heather will then add on some environmental variables. And finally, I'll close out with some connections to ecosystem scale.
It's good for the microphone. All right, so hi everyone. I'm here to talk to you guys about my project, which looked at the developmental mechanisms and phenotypic divergence in a giant three-spine stickleback population. So my project had kind of two main goals. First, to provide a detailed phenotypic characterization of a large-bodied stickleback population via comparison with normal-bodied stickleback, as well as then examining allometry and phenotypic correlation to provide insight into some of the possible developmental mechanisms behind this phenotype. So first and most importantly, what exactly is a stickleback? Well, they're a small fish that inhabit northern habitats across the northern hemisphere, including coastal marine, brackish, and freshwater environments. And they're commonly used as a model for contemporary evolution. So stickleback exhibit very extensive habitat-specific morphological variation due to specialization to local conditions in this extremely wide distribution. So for the first of my traits that influence phenotypic variation, I'm going to talk about allometry, which just refers to any sort of biological scaling relationship in which a trait changes with size. In my case, I'm looking at how does shape change with size, as seen here. And this is one of the known contributors to phenotypic variation in stickleback. Another contributor is integration, which refers to coordinated variation in functionally and developmentally related traits. This is commonly investigated using phenotypic correlation as a proxy measurement, but this may or may not be necessarily the best way, as more recent research suggests a more cautious approach here, with correlation suggesting integration rather than meaning integration. So gigantism just refers to anything with abnormally large size and is usually very ecologically or evolutionarily significant. It is rare, but has been documented in stickleback, specifically isolated lake populations in Alaska and the Queen Charlotte Islands. As seen here, this on the bottom is a giant stickleback from the Queen Charlotte Islands, and these are more normal sized stickleback. This figure also does a great job of showing some of that morphological variation. And while this has been documented, it hasn't really been studied in extensive detail. There hasn't been any of these phenotypic characterization studies. So my target population is from Sarita Lake on Vancouver Island, British Columbia. They are unusually large sized. They're about 33% longer and more than double the weight of these other local populations. They're not quite as extreme as some of those previously mentioned giant populations, but they are quite a bit larger than a normal stickleback. To illustrate this, these are a pair of 3D models from CT scans. On the top is a fish from that Sarita Lake population, and on the bottom is a fish from a place called Frederick Lake. These guys are kind of the normal sized, and these are the giants. So to do all my investigation, I did a method called geometric morphometrics. Morphometrics just refers to any sort of quantitative shape analysis, and geometric morphometrics uses landmarks placed on homologous anatomical loci, and then the XYZ coordinates of these points can be analyzed mathematically. So moving on to my actual research. I had a total of 6 populations from Vancouver Island. There were 4 lake populations, including that Sarita Lake and Frederick Lake, which will be mentioned more because it is the closest lake geographically to Sarita Lake, as well as 2 marine populations. For my geometric morphometrics, I used a landmarking scheme from Schutz et al. 2022, which split the landmarks into 3 regions: the skull, the pectoral girdle, and the pelvic girdle. For Sarita Lake and Frederick Lake, I actually produced the 3D models and landmarked them myself. And the remaining 4 populations, I used data from that same Schutz et al. paper. My morphometric analyses fell into kind of four general regions. A principal components analysis, which identifies main axes of variation and helps to kind of group the fish, followed by a Procrustes ANOVA, which looks at the impacts and interactions between population, size, and sex, and how these affect shape. I then did pairwise comparisons of all the populations looking at mean shape and allometric slope. And finally, I looked at phenotypic correlation in both size and shape. So starting off with absolute size. As you can see here, again, Sarita Lake is the purple ones. They are the largest lake population in every single region examined, and they have the largest skull and the largest pelvic girdle by far. We can also see that the marine fish do tend to be larger. In terms of shape, Sarita Lake did exhibit unique shape morphology in every region examined. And on the PCA analyses, which you'll see in a moment, there was a tendency to overlap with Frederick Lake. So this is one of those PCA analysis figures. As a quick outline of what you're actually looking at here, each size or each axis will represent a component of variation that contributes a certain percentage to the overall shape variation. In this case, PC1, the largest component, and PC2, the second largest component. So as you can see here in the skull, Sarita Lake is a little bit distinctive up there, but it generally aligns with the freshwater fish. Now we start to see quite a bit more divergence in the pectoral girdle, where Sarita Lake is now occupying a much more distinctive area compared to the other populations. And this becomes even more prominent in the pelvic girdle, where Sarita Lake is now in a almost entirely on the other side, as well as overlapping a little bit more with that Frederick Lake, which again is the closest population. Moving into the allometry, as a quick disclaimer for some of these figures, this shows the actual distribution of regression scores with size, and there is quite a lot of variation within every population. So the trend lines, as you see here, kind of over-exaggerate the amount of variation present. That being said, in the skull at least, all of the pairwise analyses found that Sarita Lake had a distinct allometric trajectory from every population, except for, again, that Frederick Lake. Moving into the pectoral girdle, this is also where you see a lot more of that exaggeration. As you see, these trend lines look very different, but the pairwise comparisons found no significant differences with Sarita Lake and any other population. Onto the pelvic girdle, this one proved a little bit more of an interesting analysis, because the ANOVA for this region actually found there was a significant interaction between sex and size and population. So unlike the previous studies, the pairwise comparison was conducted by splitting up sexes. So male of one population, male of another population, female, male, etc. In this case, there was not quite as many differences as the skull, but more than the pectoral girdle. And most of these differences involved a female Sarita Lake versus males from some of the other populations. Onto the correlation studies. In terms of size correlation, every population had significant correlations between every region. Comparing these correlations, Sarita Lake had differences in the skull and pelvic girdle and pectoral girdle and pelvic girdle correlations with the marine fish and Klein Lake, which does actually make sense because if you think back to those box plots, Klein Lake was by far the smallest pelvic girdle, and they differed from the marine fish in every region. Now, shape correlation has some of the more interesting data happening here. There was a significant correlation between the skull and the pelvis and the pectoral girdle and the pelvis, but no significant correlation in shape between the skull and the pectoral girdle. Sarita Lake was also the only population to exhibit two significant correlations. Furthermore, they had the largest skull pelvic effect and the second largest pectoral pelvic effect. So what does this actually mean? While there was a distinct whole body shape phenotype, the developmental patterns seem to mostly be restricted to the skull and the pelvic girdle. The pelvic girdle is particularly interesting here because it's both extremely large and is involved in those significant correlations. Pelvic girdle morphology in stickleback is commonly associated with predation pressure, as it's a key part of their kind of defensive apparatus. So as you can see here, this is a bottom view. The pelvic plate acts as belly armor effectively, It also braces these pelvic spines, which can be erected during a predation event. And the ascending process, which comes up here, helps to actually brace the dorsal spines as well. Reduction and loss of this girdle has been documented to be associated with an absence of predatory fish, while higher levels of predation seem to favor a much more robust girdle. So the strong correlation with the pelvic girdle and the rest of the body regions could mean that, assuming this indicates a strong integration, predation-induced changes in the pelvic morphology could then subsequently induce changes in the skull and the pectoral morphology through that integration. Now, a predation hypothesis is consistent with a lot of the literature that does exist on giant stickleback, which tends to exclusively focus on predation. One experimental study on these stickleback found predation failure from predatory fish. So basically a situation where a predatory fish cannot actually eat the stickleback. Occurred at stickleback of body sizes between about 40 to 70 millimeters. Sarita Lake tend to average about 66 millimeters, so they're to the far end of that, and some are quite a bit farther outside of that range. On the opposite side, stickleback themselves are predators. So being larger would allow them to eat different prey types. This novel prey could lead to changes in the skull morphology, which would further change the overall phenotype. Finally, there's some potential role for sexual dimorphism and sexual selection in the development of this phenotype. A study on 9-spine stickleback found female-biased sexual size dimorphism, which is indicated by that size-sex interaction in the pelvic girdle, was implemented in the development of gigantism in this species. And it is worth noting that most allometric differences with Sarita Lake did involve females, as well as there being that size-sex interaction. But this is a fairly tentative connection, but worth looking into in the future. So in conclusion, characterizing this phenotype provided valuable insight into the development of a giant phenotype in this population, with the allometric and correlation tests pointing towards the evolution and development of the pelvic girdle in particular, having a key role in this phenotype. Altogether, this provides direction for in-depth studies into gigantism and potentially other abnormal morphologies in this species. And I just wanted to shout out again, everyone in the Jaminski and Lucas and Rogers labs, everyone of the stickleback people, Kevin again for helping me with all of the stats for this project, and the authors of that Schutz et al. Paper, whose data and landmark scheme I used. Thank you.
Can everybody hear me if I just talk like this? Okay. Oh, never mind. Is that okay? All right. Hello, everybody. My name's Duncan. I'll be telling you about a totally different kind of fish, a group of fishes known as chimeras. And here's one species of the chimera here. It's a spotted ratfish. It's the only chimera that is native to Canada's west coast. And they're cartilaginous marine fishes that are closely related to sharks, to skates, and to rays. I started studying them because they have this really unique mode of locomotion where they flap their pectoral fins up and down. But as they do this, they generate this undulatory wave that will pass from the leading edge of the fin to the trailing edge of the fin. That white line here right now is outlining that wave that's going to happen as the fish flaps its fins up and down. So you can see that as the fish is flapping its fins, there's that wave that's traveling from the front, the leading edge of the fin, to the back or the trailing edge of the fin. And understanding an organism's morphology and mechanics leads to an understanding of their ecology. So that is why I looked into the shape of the pectoral fins and how this changes swimming dynamics and movement. So there are 3 families of chimeras. There's the plow-nosed Calorhynchidae, the long-nosed rhinochimeridae and the blunt-nosed chimeridae. And not only do their body and their snout morphologies differ, but their pectoral fin morphologies also differ. So you can see that Calorhynchus and Chimera monstrosa have this more slender, high aspect ratio fin, whereas Rhinochimera pacifica has this more low aspect ratio, broad, blunt pectoral fin. And to quantify the variation in shape between these pectoral fins, I also use geometric morphometrics. So I went to museums in Chicago and San Francisco, and I took pictures of the specimens there, and I put points on spots that were easily identifiable between species. So where the muscle and the fin connects at both the front and the back, and then also at the tip of the fin. And then I placed curves on the leading and the trailing edges of the fin. Now, I was thinking that ecological factors might place selection pressures on fin shape and contribute to the diversification of these chimeras. For example, that spotted ratfish I showed you earlier, that was the scientific name's Hydrolagus colei. And then there's another fish called Chimera monstrosa. And then all three species from the Calorhynchidae family, they migrate to shallow wave-swept waters to spawn. And they're the only 5 fishes out of the 55 known species that do this. And I'm thinking that these fishes might have pectoral fins shaped to migrate efficiently. So after doing that geometric morphometrics analysis, come up with this morphospace, which is really just showing you how different the pectoral fin shapes of these species are. So I have 17 chimera species here from the 3 families. For example, Chimera jordani's pectoral fin shape is up here, where Chimera phantasmas is down here. Species that are closer together have more similar fin shapes. So Calorhynchus capensis and Calorhynchus calorhynchus have much more similar fin shapes than the 2 I pointed out earlier. And then the yellow and red and blue polygons represent the 3 families. So Chimeridae is in blue, Calorhynchidae is in yellow, and Rhinochimeridae is in red. And I chose to model 3 of these species to see how pectoral fin shape influences swimming dynamics. And I did this based on family, and I did this based on PC1, which is just a description of the greatest axis of shape variation. So shape varies more on this axis than it does on this axis. And this axis accounts for 44.3% of the variation in shape. So I chose Chimera jordani from the Chimeridae family all the way on the negative end of PC1. I chose Rhinochimera pacifica from the Rhinochimeridae family, and that's more on the positive end of PC1. And then I also chose Calorhynchus calorhynchus from the Calorhynchidae family, and this is a more central, a species with a more moderately shaped pectoral fin. And this is what those models turned out like. So they're made of this flexible silicone rubber material called EcoFlex. And I traced those fins with geometric morphometrics, printed some molds on a 3D printer, and then filled them with this silicone rubber. You can see that Chimera jordani, all the way on that negative end of PC1, has this high aspect ratio, slender fin with a concave trailing edge. The trailing edge is curved inwards. Whereas on the positive end of PC1, you have pectoral fin like Rhinochimera pacifica, which is blunt, low aspect ratio, and has a curved outwards or convex trailing edge. And then you have that more moderate fin shape with Calorhynchus calorhynchus. And to study the differences in these fin shapes and how they affect swimming dynamics, I built a fin flapping robot to mimic the pectoral fin movement of chimeras. That video will play. That's all right. It doesn't look like it's going to play, but what happens is there's a couple motors on the top that flap this fin back and forth, and then I'll get into some fancy ways to measure the water flow around these structures. really all you were going to see it do was turn around to the front side. You could see the fin being flapped and forth, and then it was going to zoom into the motors on top. And there's one that controls the amplitude of the fin and the speed. And then there's another one that controls the pitch of the fin, which is just the angle that the fin is flapping through. And although controlling a robot is a lot easier and a lot more controlled than controlling a live organism, I had to do a lot of fighting against the code. It was a huge pain, but eventually I was able to figure things out and get on to the data analysis and the data collection. So I'm not really sure how familiar any of you are with PIV or particle image velocimetry. So I'm just going to give a really hopefully easy to understand summary of what it is and what I used it for. So what you do, I guess first I should say that this would be a left pectoral fin of one of those chimeras, and it would be flapping up and down this way, and the fish would be moving to your left. And then I dumped a bunch of tiny fluorescent particles in the water, and I shone a laser at the water through two lenses to split that beam into a sheet, and then the sheet will illuminate all those tiny particles in the water. You can kind of see them here. They look like dust specks. And then you can film this at really high speeds to track where those particles are moving. And because they're in the water, you can kind of see how the water is moving around the fins. The reason you don't see any behind the fin is because the fin itself casts a shadow. So you just can't see the particles there. And then when you plug it into a computer, it'll analyze it and it'll give you something like this. So the arrows represent velocity. For example, this point here is moving up and to your right quite quickly, whereas the point over here is still moving up and to your right, but it's going at a much slower pace. And then the red and blue represent vorticity, which is just a fancy word for how swirly the fluid is. So red shows counterclockwise vorticity, which is rotation of the fluid in the counterclockwise direction. And then blue shows positive vorticity, which is rotation of the fluid in the clockwise direction, or sorry, negative on blue, positive on red. And the intensity of the color shows how quickly that vortex will be rotating. So that vortex here is darker color, it's dark blue. It would be moving quite quickly in the clockwise direction, whereas this vortex here would be moving relatively slowly in the counterclockwise direction. And when you put that all together in video, it looks like this. And there are a couple important things to note in this video. So first, as the fin moves up on the upstroke, it creates this negative pressure region underneath it, and water flows into this region upwards into the fin. You can see all of those arrows are pointing upwards into the fin. And then as the fin reverses direction, so it's gone from upstroke, now it's switched direction, it's on the downstroke, that water with all of that momentum can no longer flow upwards into that region. So instead, it's forced around the trailing edge of the fin. And you can see that the fluid is starting to curl around the fin here. And there's that red vortex beginning to form there. As the fin continues on the downstroke, that fluid will continue to curl into that region, and that vortex will grow larger and stronger. And just as a vortex is created at stroke reversal on the top of the upstroke, a vortex is also created at stroke reversal at the bottom of the downstroke. So that's this blue vortex here. And something really interesting happens when two vortices of different spins interact. They'll create this jet of fluid between them. So this red one's rotating this way and the blue one's rotating this way, and they'll push this jet of fluid between them. And that causes this propulsive thrust, which would push the fin forward. And if there was a fish attached to the fin, it would also push the fish forward. Okay, I will remind you what these three fins are. So this was the more moderately shaped Calorhynchus fin. This was that slender high aspect ratio Chimera jordani fin. And this was the low aspect ratio Rhino Chimera pacifica fin. And you can see that the Calorhynchus fin produces two really nice vortices, a really nice jet of fluid between them, and it is generating the most effective jet. Whereas Chimera jordani doesn't space the vortices appropriately. So instead of interacting to create this jet of fluid, they crash into each other and annihilate one another. You can see that that red vortex here is cutting that blue vortex in half. Eventually, if this were to be a video and it was to keep going, it would kind of dissipate into the water. And then Rhinochimera pacifica does create appropriately spaced vortices, but they're a lot weaker, so it doesn't create quite as effective propulsive jet as Calorhynchus does. I also backed this up with some force torque measurements with a sensor, and it did show that Calorhynchus created the most thrust out of these three fins. Interestingly, Calorhynchus is the only fish out of these three to migrate, which supports the hypothesis that fish that migrate might have pectoral fins that are shaped for migration. And in summary, the fin shapes differ between families. Calorhynchus generates the most thrust. Fin shapes may aid in effective migration. And this research could provide insight into the ecology of chimeras and could also lead to the development of novel underwater vehicle propulsors. Thanks.
Let's grab the right talk here, sec. This one, nope, that one's Sarah. It's Duncan. Sorry, folks, give me a second. This one. There we go. All right. This.
You select the slides. Is that where you want to start?
Yep. Well, this is the same slide. But yeah. All right. Thanks, folks. Sorry for that. So I'm Heather Jemnitsky, and you've seen this slide before. And so what we're going to do now is bring this back a bit into this ecomechanics space. So you've seen some stuff about the detailed looks at morphology that we do. You've seen some things about the detailed look at biomechanics that we work on. So now we're going to think again about this, putting these two things together, right? So functional traits, morphologies, these things that we understand, plus the environmental variables. What does that do for us when we're thinking about the animal in the space and how the evolutionary processes that are acting on it how that might actually work? So I'm going to bring you back to this one again. Sarah gave you a nice introduction of these little fishes already. But one of the things that's interesting about them, as she mentioned, they're a model for contemporary evolution, and they do these rapid transitions from marine to freshwater habitats. So Sarah was comparing both these marine and freshwater animals to each other. They can do that. They're ancestrally living in the ocean, and all the time over their history, last 10,000 years, they hop into a freshwater environment, like a salmon, something spawning, it goes upstream, goes freshwater, and then decides to stay there. Whether it stays there for some ecological reason that's sort of local and proximate, or whether they get trapped there, as has happened in the last ice age, they will stay in the freshwater space and adapt to that space. So we have a number of projects in our lab, some of which you've already seen, about what happens when they do that. Shape changes, size changes, changes in thermal tolerance. If you're in a little tiny freshwater body, you're subject to temperature fluctuations that wouldn't be there in the marine space. Growth and development, gene expression patterns. There's all kinds of strange things happen when they do this. One of the things I'm going to focus on, or I guess the main thing I'm going to focus on here today, is when they go into freshwater, and again, Sarah alluded to this, there are often changes in the predation regime that they're exposed to. When they arrive in these places and stay there, they lose body armor. So as marine fishes, they've got these beautiful bony plates embedded in their bodies all the way down. So when you think of a fish, you often think of scales. These guys actually have big bony armor plates all over their body. So very cool. When they're marine environment, they look like this. As they transition to the freshwater environment, they lose those, and they end up with 3 or 4 of them left, and all the rest of the body is wide open. That happens every time they go from marine to freshwater environments. So there's some pressure that changes in that space. We know that about 70% of the variation in this body morphology and this armor morphology is controlled by a single gene, this ectodisplasin gene. There's also a receptor for that gene that accounts for another 10, 15% of that variation, but there's still a little bit of noise there, which is kind of interesting. And then there are additional body shape changes that go with this, changes in sexual dimorphism patterns, all kinds of things that happen. But keep this in your mind for now. You go from fully plated, fully armored to no armor every time you go to freshwater. What's pushing that? The literature is full of theories about this, some of which relate to osmoregulation. That ectodisplasin gene is a physiological gene with effects on various physiological processes. Some of the thought is around calcium limitation. Maybe in a freshwater environment, you just can't build bones. There's not enough resources to do it. Could be that. Maybe, and we know that there's a juvenile growth advantage for these ones in freshwater, so the babies grow faster. Maybe the overwinter survival is better. Maybe if you're not building this bony armor case, you do better over the winter. Other ideas are around predation regimes, as I said, or swimming ability. If you've got this row of bony plates down the side of your body, that's going to impact your flexibility. It's going to impact your hydrodynamics, all kinds of things. So maybe it's this. So the question we've recently asked in our lab along this ecomechanics framework is, what if it's both of these things? So if the predators are changing, maybe what you have to do to get away from the predators is changing. If the predators are changing, maybe the ways in which you protect yourself from the predators are changing. So in a marine environment, whatever's hunting you there, maybe you actually need this full body armor suit because they're going to catch you. So what you want to avoid is getting actually crunched and eaten. Whereas if it's something different in a freshwater environment, maybe what you need to do is get away. You don't need the full body suit because nobody's going to catch you as long as you can swim fast enough to get out of the way. So we wanted to ask this question, and the challenge that we have, of course, with the ecomechanics model, and some of you might be thinking about this already, is like, okay, that sounds cool, but how many variables are there when you actually go out into the real world and try to measure something? It's a huge problem. So with this animal, we've been very fortunate to find a couple of instances, and I'm going to talk about one in particular here, where we find an animal, a population of these fishes that is actually in the process of transitioning from a marine to a freshwater context, not because the fish has gone somewhere else, but because the environment has suddenly changed. So Stanley Park, downtown Vancouver, many of you I'm sure have been there. This is Lost Lagoon. You can go for a nice little stroll around there. There's a little paved pathway. It's all very nice and urban and whatnot. Turns out this was actually connected to Cole Harbor until 1916. When they built the seawall across here, they basically put a dam there, and this little bit of water was isolated from the ocean. So we went from an ocean space to a freshwater space. By 1930, that was completely closed off. So the fish didn't go anywhere. The environment suddenly became freshwater on them. And so that started to force an evolutionary transition to happen. And I will invite you to take note of the timing here, 1930. So we're, less than 100 years, human lifespans. So we went in there and we had a look what's actually going on in there. We find all three genotypes related to that ETA gene that I talked about, almost evenly distributed in that space. So if you're in the marine environment, you have one ETA genotype that drives the high plate form, the full body shape. In the freshwater, you have a low plate version of that gene, no armor. In this lake, you got all three. So you got a mix of genotypes. So something is happening here already to the allele frequency in that driver gene, less than 100 years. So that's kind of neat. And then we took a look at the phenotypes following the same type of mechanisms that Sarah used, talked about earlier. I can come back to this and talk about it later if folks have a lot of questions. All I want to show you here is that we found all three of the different plate morphs. So we found the full morphs, the marine ones, we found the freshwater morphs, and we found a bunch of transitional forms. They've lost half their plates. So morphologically speaking, we are making this transition from marine to fresh in this space. And interestingly, if you look at full body phenotypes, you can see that we've got two freshwater comparison groups over here. We've got two marine comparison groups up here, and then our focal population here. So they've changed from a marine full body shape, but they've not got real close to a full freshwater body shape either. So they're on the way. They're not there yet. So again, not the focus of this talk, but just to convince you that this is actually a location where these animals are, they're doing the thing right now. They're going through that evolutionary transition on a contemporary scale in an environment that we have control over, in the sense that all three types of morphs are in the same space And that's a natural experiment. So that's very cool. So we went and did some biomechanical work, kinematics work really, on those guys. So the study design that Aspen used, and this, again, a former master student of mine, fantastic job of work, collected a group of fish from that pond. We crossed some of them and we kept some of them. Some of the adults that we kept, we did a series of experiments on. We looked at phenotype. We looked at escape response performance. So C-start escape response, and I'll show you what that looks like in a minute. The ones that we crossed, we then did experiments, same set of experiments on the juvenile offspring. And then we split them into a saltwater group and a freshwater group and tested them again as adults. So what we're trying to ask here is, first of all, what are the actual guys that are in there now doing? Then we wanted to ask if there's selection pressure on your swimming capability. Is it the juvenile stage where we see that or the adult stage where we see that? Because one of the things we don't often take into account when we're thinking about evolutionary change is, where does it hit? Is it hitting the babies so they never grow up, or is it hitting the adults so they never reproduce? So we tried to capture both of those things. So the kinematics that we did here, this is C-start kinematics. There's a fish, stickleback fish up there. This is my student's hand with a little stick. This is really sophisticated stuff, right? We've got a fish in an aquarium. Aspen is gonna bother the water behind the fish. What you're gonna see the fish do is form its body into a C shape and then push off across the screen. That is a standardized escape response for many, many kinds of fishes. And so what we can then do with that is we can calculate things like velocity, like acceleration, like distance traveled. So if I have better luck than Duncan did, there you go. You can see the stick is going to come down. She's just going to tap the water. There's the C forming, and there's the fish blast off. Incidentally, another student of mine is doing some work on a similar experiment and has found that these fish have an astonishingly high speed associated with this. It looks very pedestrian in this image, but these fish they're pretty good at this. So we have them do this a number of times. We calculate maximum performance for different variables. I'm going to talk to you today only about total distance traveled for reasons of time. But just to remind you, this is the question that we're asking. So does escape performance, as we measure it here with a C-start kinematics, does that drive the adaptation to freshwater habitats in three-spined sticklebacks? In other words, you don't want to get caught because you haven't got the armor. So is it going to be different? So here's what we found, some really interesting results here. So these are our wild-caught adults. So these are the first ones that we just grabbed out of the pond, took them to the lab, and tried them out. So what you'll see here is, again, in purple, these are our ones with the genotype for the full body armor. The blue, green, that's supposed to be teal. They're the heterozygotes, and then our low-plated ones are in yellow. So females on the left, males on the right. What you can see here is that there's a trend towards greater total distance traveled in the females. Remember what Sarah said in her presentation, that some of these traits seem to be driven, there seems to be a sexual dimorphism here, driven by females in the morphology side. Could it be that this is also something happening here in the performance side, again, driven by females? When we go and look at our juveniles, so these are the ones that we crossed before we put them in two different habitats. We just crossed them, raised them up sort of part way to sexual maturity and measured them here. You can see that this is actually gone. There's no anything interesting to report on this figure. These yellow ones, again, these would be our low-plated fellows. And you can see there that there's really no difference across the juveniles. So this would indicate to us that whatever is influencing this is not happening at the juvenile time point. So then we went ahead and raised them either in saltwater or freshwater with replicate families so that we didn't get, occasionally you get sort of like a superhero family doing things. So we had several replicates of this to try and account for that. And that's what you'll see here. Saltwater on the left. So 3 families now that we measured, these groupings is a family. Freshwater raised adults on the right. So this has us really excited because what we see here, what we think this is showing us is that our low-plated animals, the yellow ones, raised up in freshwater have a greater total distance traveled than they do in saltwater. So we think that this is telling us that there's that effect again that we saw in the first results slide that I showed you, is that there is something here indicating to us that if you have a low-plated allele, or 2 in the case of these yellow guys, and you're in a freshwater environment, you're better at escaping. So to kind of bring this back into a something that we think makes sense. In this transitional population that I've shown you, the females seem to be maybe, and that result wasn't completely, I'm not totally convinced by that. I don't want to oversell it. It's interesting. We need to do some more work on that. But it would seem that there is a sexually dimorphic feature here. It's the females that are capable of a greater total distance traveled if they have one low-plated allele or more. In those adults that we crossed, when you have two low-plated alleles and you're in the matching environment, so in the freshwater environment, you're better at getting away. So that's kind of neat. That kind of implies that there is, in fact, a performance advantage to having that low-plated allele. We were able to test that in a controlled environment, a controlled natural environment, which you don't get all the time. And so for us, this is an example of where you can do this. You can take environmental variables, functional traits, combine those into a model. And now we're starting to see something that might make some sense. So all those different examples I gave you in the literature, osmoregulation, calcium metabolism, overwintering, maybe it's not those things. Maybe we actually have some evidence here for what it is in that wild condition or in the natural condition. So I'm going to stop there and pass it over to Kelsey, who's going to talk about ecosystem level attacks.
Okay, so I'm going to close this out by giving us a quick run through of ways that we're thinking about stuff at the ecosystem level. The idea here is that when you're thinking about real environments, there's just so many things going on. They're very complex and have complex conditions and histories. So just thinking about a tide pool, for example, because that's close to where you'd find a stickleback. If you're thinking about performance, you might have temperature effects on metabolism and muscles. You might have swimming performance effects due to turbidity. So in a predator-prey encounter, for example, fish use their vision quite a bit. You also have issues with flow and turbulence that could be related to sensing and stability. And all of this is happening at the same time as escapes and detecting predators and everything else happening in the environment. And then there's also the one that we usually forget about, which is that for freshwater fishes, you do have to think about repopulation pathways after glacial retreat. So with all of this in mind and our ecomechanical framework in mind, we're still we've got some convincing results that environment and functional traits are kind of coming together to create mechanics that may have fitness implications. But there's just so much stuff going on that how all of this fits into a larger ecosystem scales and the role of biomechanics on the ecosystem scale has been in my mind over the last few years. And so when we're thinking about this in terms of freshwater fishes, we often think about fishes as classified as in one of two extreme shapes. At the top here, you see the more streamlined form, and then that contrasts with this bulkier form on the bottom. And the big idea here is that the ones on top are more streamlined. You expect them to be, and so we just naively say, hey, these guys are probably better, more efficient swimmers. Whereas the ones on the bottom have larger structures, may be able to generate forces and torques very rapidly. And so we tend to think about them as unsteady swimmers. I'm going to note that very little of this has actually been tested, even though we have been saying it since the 1980s. Now, when you take that into freshwater habitats, we tend to map this onto different habitat occupancies, especially thinking about lake areas. So in a lake, you may say, hey, those streamlined forms, I might expect those to be in the open water environment. Open water, lots of space, got to move around really far. That makes sense. And then when you're thinking about a lake, you may say, hey, maybe we'll have more of those unsteady specialists on the bottom or on near shore environments because there's more complexity, there's rocks, there's fallen trees, all that kind of stuff. But we say all of that, again, very little has been tested. And there's all of these other factors that might be influencing these habitat uses. So I found this really interesting model system that's Michigan inland lakes. So the state of Michigan among the Great Lakes, just for reference, Toronto would be about here-ish. And so this is a really interesting system because there are about 6,400 inland lakes in the state of Michigan that are greater than 10 acres in surface area. So this is a big lake, one that you'd have multiple boats and recreational fishing on. These lakes span several environmental gradients, especially climate with the north-south axis. There's also an east-west climate axis due to lake effects from the Great Lakes. And these gradients also span size, fetch, which is the maximum uninterrupted distance that wind can blow over, and so is related to wave energy in the environment. Sea levels of human influence, different glaciation patterns, and so forth. Also, this system is very well surveyed, both in terms of the fishes themselves. There's this, as well as in terms of fishing. Sorry, the word is catch per unit effort, but that is a measure of abundance in the lakes. So both fish abundance as well as morphology are pretty well surveyed in the system. So just to verify that this gradient between steady and unsteady actually exists, again, this has all been, we've been saying this, no one's actually tested it. I went to the Michigan Museum of Natural History, photographed a bunch of specimens, put landmarks on them, measured a bunch of things. You've already seen this a bunch of times. And when I threw this into a preliminary PCA with some of our more extreme shaped fishes, just to get a sense of the space here, we do actually recover the steady versus unsteady continuum, particularly on the first principal component axis here. So this shows you the shape variation among these species. The first principal component here represents about 40% of the variation. And we see a lot of difference in streamlining, with streamline forms over here and bulkier forms over here. We also see on PC2, which is about 24% of the variation, a lot of shape metrics that we tend to associate with things like caudal fin use, steering capabilities, so pectoral fin size, pelvic fin size, and so forth. And on both, we also see some diet and stability factors as well. So that's cool. We know that this morphological gradient exists. And so how does this actually map up to habitats? And the answer to that is we're still working on it. And that's because these species abundances are wildly complex. Now, for the 8 species in the preliminary PCA, what I've done here is just mapped out their abundance across the state. So that's all you're looking at, just straight abundance data. Because fish abundance is going to depend on what gear you are using to catch the fish, and it is hard to standardize gear measures. So how do you turn a per lift into a minute of electroshocking if you're using a net versus an electroshocker? So to get rid of that problem, I've standardized, for the sake of these charts, the gear to the gears match maximum catch per unit effort. So the largest dots are 100%, the largest abundance that you would get with that gear, and the smallest dots are the smallest abundance you would get with that gear. And again, this is just to aid us with visual comparison. And then we have the different gear types and different colors. Now, again, looking at these plots, I bet if you're squinting at these right now, you're saying, like, where is the trend? And yes, that is exactly the problem. We're seeing that these abundances are really, really complex. And so in order to actually get at what these maps, we have to use some species distribution modeling techniques. Now, I'm not going to go into a huge amount of detail on the model because it's kind of boring, but the idea is that we modeled species abundance based on species, gear type, climate variables, water quality variables, and a number of other variables that are likely to be related to swimming performance. We also included two-way interaction terms between species and pretty much all of the other variables. And once we ran this model, I want to highlight a couple interesting findings that we've seen. So after we've accounted for climate and water quality, which no matter when you look at a fish, where you look at a fish, those are going to be the #1 most important things for species abundances. So we've included them in the model, we've accounted for that, and now putting that aside, what's left? We find that connectivity is an important variable. So this is a categorical variable that measures how connected lakes are to other lakes. So a lake may be an isolated, independent, like pothole type of lake, or it might be a lake in the middle of a river chain. And so you would think that a lake in the middle of the river chain has more opportunities for fast, streamlined swimming fish to be moving in and out of the lake through a river where you have to swim against flow. We also see a significant effect of fetch. So this is our wave energies. So that suggests that fish being able to stabilize might be an important factor for species distributions. And we also see an effect of submerged fallen trees per km shoreline. Now that one's a really interesting one because from a biomechanical standpoint, we'd say, hey, that's a complex environment we got to steer around. We need some unsteady swimming specialists here. But I also want to point out that this is important substrate for spawning for many species of fishes. So we have to do some further analysis on that. The idea is that we've got some unsteady specialists in the data set that do use that substrate for spawning and some that don't. And so we're going to look to see if we see similar trends between the two. And now, in order to finish this project and really get at that landscape piece, what we really want to do is integrate that morphology data with these species distribution models. And you can do this with the modeling technique called fourth corner modeling, which essentially lets you ask the question, does morphology correspond with gradients and connectivity, fetch, and submerged trees? And so we hope to do that in order to better map all of this biomechanical stuff onto landscapes to get a better sense of how it fits into the bigger picture with all of these other like temperature and climate variables. And then because it's not yet possible to do a fourth corner analysis in a phylogenetic framework, we hope to repeat this type of analysis over multiple landscapes to see if the trends that we're seeing here is something about just this set of fishes or these lakes versus something that we can say more generally about ecomechanics. And so with that, we'll say that adding environmental variables to predictive models that include functional traits, we do think this is allowing us to better characterize evolutionary patterns in fishes. And I will leave that there and offer the floor up to questions for the entire panel. Thank you very much.
Awesome. Thank you so much. So I don't know who. To the group. So we'll get started. Yep. You have a question?
When during the life cycle do these plates develop?
So excellent question for the Zoom folks. When do the plates develop during the life cycle? They actually develop quite early on. So we start to see cartilaginous precursors of them along with the rest of the skeleton. So they're tightly integrated with the skeletal development. I would say just before sexual maturity, the plates are pretty much complete.
Do you see any difference in getting into adulthood between the two, the one with the more plates, the one with the...
Sorry, can you say that again? Any difference between
adulthood in time?
No, so excellent question for the Zoom folks. Is there a difference in reaching sexual maturity in the plated versus the low plated ones? No, the timing is approximately the same. There are other factors that influence how quickly they mature, but it doesn't seem to be an effect of habitat or plates. Yeah.
Andrew?
The stickleback size in Surreal Lake, do they have a higher predatory pressure there than other lakes?
Yeah, so again, for the Zoom, that's is there a higher predation pressure in Cerrito Lake compared to the other lakes? So that's kind of the interesting thing is I'm almost working backwards because I have the fish and I have the size data, but I don't really have any ecological data. The one paper that did do some of that size measurements was actually studying freshwater mussels. But they did find there was a difference in parasitism regime there. They didn't look into it because it wasn't relevant to their focus. But there's something going on ecologically that hasn't been looked at.
I also have a question for Sarah. So in your, like you were looking at the size differences, do you think that in aquatic species that you see, would you expect that you would see greater variability in size in comparison to terrestrial species due to I'm imagining if you're a terrestrial species and your size is limited more by gravity essentially and is restricting your motion, an elephant can only get so big and not snap its bones. In an aquatic species where there's buoyancy, would you see more variability and being able to prioritize more of those ecological factors as opposed to mechanical limitations?
So I don't know exactly. I do know that you see a lot more variability in freshwater species than marine, at least for the stickleback. I would assume at the very least you can get much, much larger for your relative body size and relative proportions in aquatic environments. I don't know if that hugely affects variability, because I don't know if you can necessarily get smaller in some environments than others, but you can certainly get a lot larger because you don't have to worry about gravity anymore. It's like stuff like the blue whale.
Right. Other questions? Yeah.
For Duncan, in human swimming, we know technique makes a big difference. And with your different fin forms, is there a possibility for subtle differences in the way the fins are used to create different flow patterns?
There definitely is. There's some preliminary research that was done at Friday Harbor Labs in Washington, where these fishes are also found. And it looks like, so what I didn't go into at all is that maneuvering, when you can undulate your fins, you can typically be good at maneuvering. And when you flap your fins, you can be good at efficient cruising. And it looks like the spotted ratfish relies on undulation when it's maneuvering, and it relies on flapping when it's trying to cruise and travel long distances. So I definitely think there are also probably small differences in shape that could lead to performance differences, and small differences in kinematics that could lead to performance differences as well.
Yeah, the fish you showed, Chimera,
yeah.
I guess. When you showed that video, it looked to me more like a bird flying rather than a fish swimming. Is there a similarity between the fluid dynamics in a bird flying compared to what you have there?
So they actually call this kind of swimming and some of the stuff that skates and rays do, flapping flight, after birds, because people are like, wow, this looks a lot like bird flight. And I think there are definitely some similarities in all organisms that are moving through fluids, whether that be insects through the air or birds through the air, or even birds through water or fish through water, mammals through water. They're generating these vortices and using them as a propulsive mechanism. So there are for sure similarities, but I'm not well versed enough on other groups to really go into detail on that.
Regarding the fin, did I see that correctly? There's a tiny muscle at the bottom of the fin, and then the rest is just passive. Is that correct?
Yeah, so there's a big difference in these fishes than in bony fishes. That muscle is all the pectoral fin muscle concentrated right at the base of the fin. And it doesn't look like these fishes have any active control over the surface shape of their fins like like bony fishes do. Yeah.
Other questions? Yeah,
a couple more questions. So I was inspired by your robot, and actually the question I had for myself, oh, is he measuring the force? Is he measuring force? And then you said you were. Do you measure the propulsive force only, or do you measure any lateral forces as well?
So I used a 6-axis force torque sensor. So it was all 3 forces and torques in all 3 directions. And the thing that stood out the most was definitely the thrust, but we also did measure like yaw and pitch and lift and all those sorts of things.
And that, if I saw it correctly, that was measured in still water, not flowing water.
Right, yeah.
So what would happen if you swim upstream or downstream? Would the patterns be retained that you show for the animals? Or
I'm not really sure. I think if you're swimming against flow, you'd need to have more energy input, more thrust to go anywhere. And if you were swimming with the flow, you might need to put in less. But I only looked at steady flow, so I'm not entirely sure.
And then I was curious, I think it was Heather talking about the lagoon there, the 100 years of isolation. How many generations is that?
So for these fish, it's probably 50, roughly. We don't know for sure. They're extremely variable. So to the other question that was asked as well, their reproductive cycles are heavily influenced by photoperiod. And so when we have them in the lab, we can produce 2 generations a year, sometimes more. In the wild, it usually takes them a couple of years to fully be ready to be reproductive, and then how long they live, we don't know. So 50 is probably a reasonable guess. So for a vertebrate, that's quite quickly, quite fast to see, you know, gene frequency change or allele frequency changes at the rate that we're seeing them.
Then for the escape maneuver, you know, you just showed the distance traveled, but I was wondering, did you kind of normalize the size?
Yes.
Yeah, that was normalized.
Yeah, they were standardized to standard length. Yeah, so the body length of the animal. Yeah.
And is that the primary measure of escaping a predator? How far we go?
So this is a good question. I might even pass this one over to Kelsey. So questions about how we interpret this type of kinematic information from fishes. There is no real sort of gold standard measurement for what you might use. So people have used acceleration as a possible. People have used maximum velocity and distance traveled. So for us, the distance traveled data gave us the most interesting picture. And so that's the one that I presented here. The velocity data was more complex and the acceleration data equally so. Difficult to pick out what's really going on there. But I don't know if you want to comment on C-start kinematics as a
yeah, so C-start kinematics, there are a lot of variables, and which one you use really depends on the type of field you're in and what kind of questions you're asking. So for example, I'm also on a students committee who's looking at response latency as their measure, but they're interested in learning and memory, and so that latency is a signal of how, if you're learning the signals, right? But in terms of biomechanics, it is usually a mess, what you see in the literature. There's no standardized thing for C-starts really at all.
What about the time it takes different fishes to form that C and go away? Because initially they don't really go anywhere as they kind of change.
Yeah, so that one actually, there tends not to be a lot of variation in that. There is some, but not tons, and in part that's because it's a reflex arc.
So the reflex arc, if you do it as the same fish, will it bend to the same side, or does it depend on where you put the perturbers? It is
completely random. So it's actually completely random. So there were a series of studies that were presented at SICFI this year that's actually trying to answer that question, which is why I mention it, to tell you, like, this is still stuff we're trying to figure out. They were looking at whether there was any consistency on whether they put the signal on the left or the right side of the fish, and where predator was and stuff. So like if I put the signal on the left side, does the fish turn to the left or the right? You'd imagine that they'd want to go to the right to get further away from the signal. It's completely random. And so there's really no we haven't really quite figured out what's dictating that yet.
Well, random would be good, because then the other fish can learn how the reaction is.
Yeah. Yeah, so and then you start adding walls and things in, like you add a wall into the middle of the tank and it changes everything up. It becomes random in a completely different way. And then ordering of experiments starts to matter. So if they have a wall first, you get different results when there's no wall, and it's still really up in the air.
Just a question kind of to extend on. So I've seen some studies, like I think it was carp, where they found there's like different fiber type properties close to the spine in comparison to further out, and that's related to that like C formation. Is that something, this is kind of a bit out there, but is that something that's like translatable to other species, and that influences those, like the speed of the starts, or is it more like the body morphology that's more important?
So those fiber types probably do matter for sure, because it would depend. I mean, we're all familiar with fast twitch and slow twitch fibers here, I would hope, or slow oxidative fibers here, I would hope. So yes, those would matter to some degree. We're not really sure yet how translatable that is to other species. Carps are weird in a lot of ways. And so I'd be a little hesitant to say that we can apply that to every single fish. But I think it is a good reminder for us that we know a lot less about fish swimming than we tend to claim. And that's what happens when you have 35,000 species of fish to work with.
Other questions? Yeah.
So how do you define the species in the state zone? because you calculated the travel distance, what is the end point?
So yeah, so we yeah, so when we're doing an escape response test, we usually go from start to maximum point of maximum velocity, and then you look until they either stop moving or they do another C. And which the fish is going to do, it depends on the fish's temperament. You get some fish that are really flighty and they'll do like one C start after another. And others, they'll just respond once and be done. So it depends a little. There seems to be a personality factor there.
And when you provide like a single as like a finger or something, is this a change with the size of the fish, or it's always a small?
Yeah, so we use the same stimulus for everything as a means of standardizing. Yes.
There's another question. Yeah.
Related to that, did the fish realize that it's like not a threat?
So this goes back to the student whose committee that I'm on, who's studying the learning in zebrafish. And now she's just done some very, very, very preliminary work. Like we test that she's been doing to make sure that the methods work, like that level of preliminary. But what's interesting in that so far is that she does seem to see like this attenuation in response if she does like 5 escape responses within like an hour period. But then when she tests the same fish a week later, it's back to square one. So this was for her, that was a really surprising result. For me, from an evolutionary standpoint, it's really interesting because you would say an escape is a bit of an all or nothing, and if you fail, you're screwed, right? But let's imagine like you're not getting caught and you don't get caught and you respond over and over and over and over and over to a benign signal. You're now wasting all of your energy budget. You're now preventing yourself from being able to feed. And so there seems to be a bit of my guess is that there's a bit of a trade-off here where learning to ignore a benign signal is probably useful for energy budgeting. But then when you get the same signal or response a week later, there seems to be some memory thing going on there.
Any other last questions? No? All right, well, we'll thank our speakers.